Hi guys, this is Henry presenting from home in China. So this is just a test video for any of you that is interested in a genetic disease. And today we're going to talk about, for your own interest, we're going to talk about a genetic disease called Adrenoleukodystrophy. And we're going to go through first a bit of its clinical overview and then mainly focus on the genetic discoveries of this disease. So let's dig in. So adrenoleukodystrophy in general is an X-linked recessive disorder, but it's also very highly penetrant disease. And apart from that, it's also a metabolic defect because normally you will have those very long-chain fatty acids in the cell being synthesized by the endoplasmic reticulum. And those fatty acids need to then be activated and then transferred to the peroxisome for degradation and also other processes. But now, due to this genetic defect, those fatty acids cannot access entry into the peroxisome, and so basically they're stuck in the cell, resulting in the accumulation of those fatty acids in the plasma and also literally all the body tissues in human. So, but clinically, the only organs that usually being affected are your adrenal gland, your central nervous system, and sometimes the testes. So the clinical feature of this disease varied between individual and also within family. And so far, the prediction of disease severity is poor at both genetic and also biochemical level. So because it's X-linked disease, you would expect male compared to female being more easily to get this disease and also have more clinical complication for that. So as you can see here on this diagram, so both male and female will remain symptomatic at birth, but later on at school age, so for male cases specifically, you will have those males developed adrenal insufficiency, which is in your adrenal gland you will produce too little of certain hormones, and the result of this is quite symptomatic. You will have loss of appetite, loss of weight, so reduced level growth, and sometimes even loss of hair. And later on, virtually all the males will develop a myelopathy, which is a compressive injury in your spinal cord. And the earliest symptoms related to that is usually urge incontinence, so you'll have a sudden urge to urinate, and also spastic gait, which is the leg stiffness and also weakness that affects normal walking. And upon all the male cases being reported so far, about half of the males, less than half of the males have developed this brain demyelination. But we're not sure when exactly it will occur. And in general, it's the damage of the myelin sheath surrounds your neuron that results in the death of the neuron and also sometimes local inflammation in the white matter of your brain. So the result is quite devastating, obviously. So the patients got this demyelination will have cognitive, behavioral, and sometimes audio and visual issues. And the patient will will eventually die if there's no treatment offered. And the cases for female is quite simple. So most of the females will have a myelopathy after their 60s. And both the adrenal cases and also brain cases in female is quite rare. So now we need to talk about the genetics of this disease, which is the main focus. So the first set of the cases of ALD were likely to be described in the early 1900s, but there's still huge speculation about the disease properties until the 1970s, when the scientists first found the biochemical feature of the disease, which is the accumulation of the very long-chain fatty acids in the cell. And along with all those biochemical studies at the times, the first genetic observation of this disease is actually through family studies. Scientists found those the co-segregation between the disease and also a gene called G6PD. So this gene was known to locate in the terminal region of the long arm of the X chromosome, so XQ28. And that's where scientists initially thought that the disease loci should reside. And later on, through more and more family studies, and also there's increasing observation in families that the patients got ALD at the same time will have the red and green color blindness. So through further linkage analysis of those families, they will be able to map the gene, like extend the mapping of the disease loci to be closely linked to the color blindness loci. But it is not until 1993 when the Moser group in the US finally mapped this gene using typical positional cloning experiment. So let's have a look at this experiment. I know this one looks quite complicated, but when I go through it, you will know that it is basically quite simple. So at that time, what they got is a patient called patient 0. So this patient has ALD, but at the same time, it has poor ability in terms of distinguish between different colors. And when scientists zoom into the X region, the terminal region of the X chromosome of this patient, what they found is, so in this patient, it has 2 deletions along with an inverted region in the middle. So for the normal person, you will have those 2 regions. When you have those 2 regions being deleted and the region in the middle be inverted, you will have the patient's condition. And the scientists found that in the first deletion region, so the patient has this red color pigment gene that gives rise to the red color vision being deleted, which, given the patient's condition, that makes perfect sense. But when they compare also the normal sequence of this region to the other ALD patient data they got, they find no other deletions in those patients, which indicates that if there's so that's what they initially did is they set up the genetic boundary for finding the candidate loci of this disease. So it should be either the second deletion or the inverted region that contain the disease loci. And what they next did is they simply just cloned the second deletion region and obtaining 3 cosmids. And they found that the cosmids can be digested by a rare set of restriction enzymes indicating CPG islands. And this signifies a promoter region, likely to be a promoter region or transcription initiation site. So what they then simply did is they subcloned the cosmids and they obtained many probes. And through both the sequencing and also computative prediction program, what they be able to find is potentially an open reading frame and spreading those several probes. And they also deduced the amino acid sequence from those sequence, and will be able to find an 80% match between the sequence and also a non-human protein found in the proximal membrane. So now through more studies, we know that this gene is called ABCD1. So one of those ATP binding cassette superfamily, and encodes for a protein called Adrenoleukodystrophy protein, so ALDP. So now we know that, so this is just in prediction, but through more and more studies, scientists will be able to confirm the role of this gene in the disease. So the first one being described is basically a knockout mice using homologous recombination, and it generates a new mice for ABCD1, and it dies very early, unfortunately, for the mice. And the scientists found that in the mice there were those enlarged adrenal gland cells, and along with that, they also find the biochemical feature, which is the accumulation of those fatty acids, and also the reduced level of the metabolism of the fatty acids in the central nervous system, kidney, and also the skin cells of the mice. So those findings were all similar to what being found in human. And along with that, later on, there's an rescue experiment. So strategy is simple. What they did is they transfected the skin cell of the patient with a normal copy of ABCD1, and they'd be able to restore the level of the metabolism of those very long-chain fatty acids. And also at the same time, they establish an inhaler cell line. Then they will be able to control the level of the expression of those non-functional ALDP protein in the HeLa cell. So do that quantitatively, which makes more sense, right? So what they did is they, when you can see here, when they induce a level of expression, and with the increased level of increased level expression of this protein, what they be able to see is the reduced level of metabolism, and also along with that, the increasing level of the very long-chain fatty acid accumulation. So up to date, and there's a more detailed knockout mice being generated. So this time, fortunately, the mice were able to live a bit longer, and then by using a combination of different techniques, scientists will be able to suggest that it is one of those very long-chain fatty acids that probably react with the free radicals in the cell, and this generates highly reactive species in the cell. And this species induces an oxidative stress in the cell, which overwhelms the homeostasis system in the cell, and leads to cell death and also tissue damage. And as part of the result here, you can see, so the scientists, they stain the callus in the cell in brown, and so callus usually deals with the oxidative stress in the cell. And you can see here in the knockout mice, 17 months old, the brown color is quite strong, indicating a high level of oxidative stress. So those three experiments all directly suggest the role of this gene in the disease, and also might allow us to know the pathogenesis that might occur in a human condition. So now we know that this protein is a proximal membrane protein, and it is an active transporter protein that is composed of 6 transmembrane region alongside with a ATP binding domain. And so what normally it will do is it will dimerize to be activated, and then it facilitates the transport of those activated version of the very long-chain fatty acids into the peroxisome. So those acids will then be metabolized, generating acetyl-CoA, and this is what normally goes to the Krebs cycle for general energy production. So now for talking about mutations and also population genetics, I like to switch this diagram to a different one, and this one is very beautiful diagram found on the ADLD website that describes basically the effect of missense mutations when it occurs at each residue of this protein. And probably you can see here that the orange and red dots indicates if there's a missense mutation happening at this point, it will likely destabilize the protein structure and results in a reduced level of the protein, or sometimes the absence of the protein, which means no protein detected. So, and you probably can see here that the region close to the ATP binding domain and also the C-terminal were likely to be a hotspot for those missense mutations to occur. So, for the cases reported so far, what we know is the incidence of this disease is about 1 per 70,000 births, and also there are about 3,000 documented mutations so far. So among them, the majority of them are missense, And among those missense mutations, about 65% of those missense mutations can result in a disease-related phenotype. So, yeah, potentially loss of function. But for individual cases documented so far, the most frequent single mutation being found is actually a dinucleotide deletion found in exon 5. So it is the deletion of the nucleotide AG, resulting in the first affected amino acid, which is glutamate, being converted to arginine, and then the change will continue. So it's a frame shift. So the change of amino acid will continue until the end of the exon 5. So the result is quite deleterious, obviously, and there's no protein being detected, and it results in a severe form of ALD. And apart from that, all the less frequent mutations, but also still frequent ones, are all missense mutations. But we need to be aware that despite all those most frequent mutations, those ones only account for 22% of all the cases being reported so far, which means that the disease mutation is highly polymorphic, and this will absolutely add difficulty in terms of generating a mouse model for understanding all the human conditions, and also in terms of producing therapies that covers all the clinical spectrum. So as we've spoken about therapies, I like to say here that the therapies up to date are still quite limited, and there's no pharmaceutical agents for treating this disease. So what you can normally do at an early stage of disease is through an bone marrow transplant, and this allows the patients to produce normal ALDP protein, so which they acquire normal function, so it generally works, but it cannot save patients when they are at the late stage of the brain demyelination that we've just mentioned. And also, it's quite hard considering finding a matched donor in real life situation. So for the other cases, when you have myelopathy, you can choose general physical care, and also you will have the adrenal hormone therapy for treating adrenal insufficiency cases. But those can only soothe the condition of the patient, and they simply cannot stop the progression of the disease, so they're not cured. And about a decade ago, there's a first gene therapy trial on 2 kids being diagnosed with ALD through prenatal screening at the time. So the strategy of this experiment, the therapy is quite simple. They extracted the stem cells from the patient, they corrected the gene, and then they put it back to the patient. And you can see here, it works pretty well for those 2 patients. So as you can see, the white area indicates the demyelinated region of the patient. So you can see here the region for those 2 patients are quite restricted after 2 years of therapy compared to this untreated. So this is quite promising. And alongside with it, there's an investigation of on developing an inhibitor of this protein. I don't know how to pronounce it, but it might look like to be L-1. So it's a protein that locates in the endoplasmic reticulum, and is responsible for the synthesis and also elongation of those very long-chain fatty acids. So this gives us hope as well. So it's pretty much it, and I think I have to conclude here that despite all those evidences we know about this disease, there remains to be huge unknown about the clinical aspects of disease, and for example, why demyelination is occurring in the patients, and also why only certain organs are being affected by the biochemical feature as a result. And there's no comprehensive mouse model so far to fully understand the human pathogenesis, and also there's no medication for this disease. But the gene therapy shown here and also potentially the inhibitor research sounds quite promising. So there's still huge possibilities for innovative researchers in this field. Thanks for listening, and have a good week.